Welcome to our video which will show you how to set up and carry out the GCSE required practical for investigating the effect of osmosis on plant tissue. In order to carry out this experiment you will need a potato, a scalpel, forceps, a white tile, a cork borer, a ruler, measuring cylinder, boiling tubes and a boiling tube rack, a top pan balance capable of reading up to 2 decimal places, a selection of solutions of different molarities of either sucrose or sodium chloride. Today we're using sucrose, some deionized water, and the appropriate Kleeps has cards. An appropriate table should be drawn up to include the concentrations of the solutions being investigated, along with the starting and final mass and length and percentage change in mass and length. Gather the apparatus and label 5 boiling tubes with the concentrations of each of the solutions being tested, adding a tube for the 0 value. This tube is going to contain deionized water. Place the tubes in a boiling tube rack. Prepare 5 potato cylinders of equal diameter using a cork borer, taking great care to ensure the borer cannot enter your hand when it exits the potato. All potato skin should be removed from the cylinders, and the cylinders should be carefully trimmed using a scalpel so that they are all the same length. The length and mass of each cylinder is then accurately measured using a ruler and a top pan balance capable of measuring in grams to 2 decimal places. Once the length and mass of a cylinder has been recorded in the table as the original or starting length and mass, it can be added to the relevantly labelled boiling tube. This is repeated for the remaining potato cylinders and tubes. 15 millilitres of each concentration of solution is accurately measured using the measuring cylinder and is added to the relevant boiling tubes. And 15 millilitres of water is added to the 0 tube. The tubes are then left for a period of time, for example 30 minutes, or as long as possible given the time constraints of the practical session. A water bath set to 30 degrees centigrade could be used to speed up the process, or the tubes could also be left to develop overnight. The potato cylinders are then removed from the tubes one by one. They should be dried slightly by plotting gently on a paper towel to remove excess liquid. The length and mass of each potato cylinder are measured, and these values are recorded in the results table as final length and final mass. The changes in mass and length of the cylinders can then be calculated by subtracting the original values by the final values. An increase in mass or length will give a positive value. A decrease in mass or length will give a negative value. The percentage change in mass and length can now be calculated from the results using the following formula. The change in mass in grams divided by the initial mass in grams multiplied by 100. The results are plotted onto a graph showing percentage change in mass against concentration. Conclusions can then be drawn to the effect of osmosis in plant tissue. We hope you found this practical advice useful. For expert technical support, contact the Philip Harris technical team on techsupport@philipharris.co.uk or visit our blog at philipharris.co.uk/blog.